My name is Amy Sullivan, and I'm the current president of AAAR. It's my pleasure to welcome you to the 37th in our monthly AS&T lecture series. This is a new initiative for AAAR being supported by the Freelander Memorial Fund. With these lectures, we hope to be able to highlight the amazing research happening in our community, tie our journal AS&T to other activities, and also give us all a chance to come together outside the annual conference. So each month, the editors of AS&T select a high-impact paper to be presented by its authors. In addition, these lectures are being recorded and will be posted later to AAAR's YouTube channel, which you can access from the AAAR website under the Events tab. In addition, each of these lectures is being hosted by one of our student chapters. I want to thank you all for joining us and for everyone who's helped to make these happen. And with that, I'll turn it over to our student chapter from Carnegie Mellon to get us started. Thanks.
Thank you, Amy. Good afternoon, everyone. My name is Oladayo Oladeji, and welcome to the July lecture series. Thank you for the opportunity for introducing the guest speakers. So today I'd like to introduce Dr. Chan and Dr. William from the University of Miami and West Point, respectively. And today they'll be presenting on concentrating viable airborne pathogens using the virtual impactor with a compact water-based condensation air sampler.
Thank you for the introduction. Yes, so I'm C.Y. Wu from University of Miami, the real Miami in Florida. And I have our co-presenter, Dr. William Vass from West Point. And next slide. Okay, so the reason why I'm doing this is just like this person finding a needle in a haystack. We know that in the air, it's full of viruses, but also they are in low concentration. So we have shown a few examples that we did in the past, and you can see the concentration of these viruses inside even a patient room is very low. In a liter of air, just a few of them. And they are so small, and most importantly, we need to keep them alive because a dead virus doesn't do anything and nobody cares about them. And so how do you actually do that? That's the topic of today's discussion. And for today's lecture, I think we are the first one with the dual presenters. And also we wanted to be more engaging. So we will ask your participation later on. And next slide, I'm going to ask Dr. Vass to give the outline and also to invite you for participation.
Thank you, Dr. Wu. Yes, my name is Brad Vass. I am thrilled to be here to talk with you this afternoon from at least on the East Coast here. I do need to start out with a quick disclaimer, though, because I am an Army officer. I need to make sure that everybody here knows that all the thoughts and opinions expressed by the presenters here are their own and do not necessarily reflect the official policy or position of West Point, the Department of the Army, the Department of Defense, or U.S. government. So it's all us. I do want to draw your attention to both the link that's in the chat and then this guidance up here at the top. We're taking another risk doing a dual online link in our presentation. So if you go to menti.com, you've got this code here. Twice in this presentation, I'm going to ask you some questions. So in fact, the next slide is going to be your first opportunity to do that. So if you go there, and then just to let everybody get to know each other here, how would you rate your familiarity with these three topics? So you should be able to see this on your tablet, phone, computer, however you're logging in. Just go ahead and vote. And I expect probably at least on the aerosol side, we'll get some pretty strong scores. But I also wanted to thank Oladayo for that introduction. So he let us know before the presentation he's nearing his graduation, and I was at that point last year, and that's a big deal for him, and really excited for him, and thrilled that he could be here to introduce us today. Okay, so look at that. We got some pretty strong scores. As expected, maybe a little bit higher on the aerosol side than the bioaerosols or aerosolized viruses. So let's just try to fill in those gaps a little bit here. So for most of this audience already knows, when we're talking about aerosols, we're talking about just a suspension of particles or droplets in gas. But when we talk about bioaerosols, all we're talking about are aerosols of a biological origin. So they can be from a range of different sources. And we're, of course, talking about viruses in this presentation. So very small, sub-micron size, very, very small, which means that those things, as those particles get smaller and smaller, well, they can not only stay suspended in the air for a longer time, but they can also make it deeper into your lungs, into your respiratory system when you inhale them. So as we think about health effects, that's really why we're concerned about this as a field. We're looking at what are the health impacts of these things that are floating out there in the air that you can't even see. And we've been thinking about this for a while. In fact, this guy up here, Louis Pasteur, way back in the 1800s, mid-1800s, started talking about germ theory, and germ theory got its origin looking at stuff that's in the air, right? So you remember, you've all seen this in a science class. Some of you have probably taught it, but you have these swan neck or gooseneck flasks, which if you leave them intact, then the aerosolized, the airborne microorganisms can't get into that flask to contaminate the liquid inside. But as soon as you tip it, or if you were to break that seal, then that water, that fluid can get contaminated. And so this gentleman here was instrumental to showing that there are these small things out there that can get us sick. And of course, you've heard of pasteurization processes. You've probably also heard of this guy, Robert Koch. So largely compatriots, the same era, mid to late 1800s. But you may have heard of Koch's postulates, how we figure out if a pathogen is actually the causative agent at different steps in doing these scientific investigations. But he was able to find the causative agents of anthrax, cholera, tuberculosis, and Pasteur as well, working on a way to combat rabies. So this was kind of the golden age of microbiology, looking at the mid-1800s to mid-1900s. But these two gentlemen did not live long enough to see the 1918 pandemic caused by influenza virus. The number of deaths due to this virus, this viral outbreak, there were far more people that died as a result of this pandemic than died in World War I. And here we have an influenza award, some historical pictures. And the life expectancy in the United States, in fact, as a result of that pandemic, dropped by 12 years. So significant impact on the well-being of people. And as we moved as a society from that point forward, we couldn't, at this point, we couldn't isolate viruses. That didn't happen until the 1930s. And so from the 1930s to the 1960s, then we were developing these vaccines, and we ended up having this seasonal flu vaccine that many of us get today. At least in the military, we get to get every single year. But we were riding this wave of this microbiological momentum, as I've named it here. And what we all want to be happy about today is that we do not have to deal with these syringes. Look how big that thing is. My goodness. I am certainly glad I don't have to have one of those put into my arm every time I go get a flu shot every year. But what a wonderful age it was, and a time of discovery, very exciting for the world, for science in general. But as we've progressed through this era, we've run into some consistent challenges when it comes to virus. Namely, how exactly do we collect them from the air? And if we do collect them, how are we going to keep them alive so we can actually detect something that is can speak directly to whether or not people can get sick, right? Because if you have a virus that's been inactivated, it's not capable of getting people sick anymore. So the way that these things are collected, they behave like particles in the air, right? They're in particles, on particles, perhaps particles themselves. But you can use inertia, like a cyclone, to turn your fluid and collect particles from that fluid. Or you can do the same thing with a cascade impactor. Or you can do that with filters through a variety of mechanisms. But at the end of the day, when we want to look at viruses, we've got to do two different things. We've got to collect them in media and detect them, maybe using some kind of like PCR, or you can use a sequencing process, right, to say whether or not something is present. But then you've also got to be able to grow it and show that that thing that was collected was capable of getting infecting cells, right? So this is a very hard process, and as Dr. Wu mentioned, when you have things like viruses that are dilute in the ambient air, it can be tough to get enough particles into whatever system you're using to collect them, and then keeping them viable is a whole nother fight. So what we used was different samplers that used a technology, water-based condensation technology, where it would pull in these particulates and it would cool them down, and then it would pass these cooled particles through this region of warm, supersaturated region, and so that water vapor then condenses onto the particles grow, and they're now within this nice little protective shell. You siphon off some excess moisture, and then you collect them into this medium that is designed to keep viruses viable. So this is the BioSpot-VIVAS, which will show up again later. So we're doing all this work collecting things from the air, but at the end of the day, it's still difficult to collect and detect viable virus. So what we do, and folks, you probably remember this from the COVID days, we're really relying on things like contact tracing and quarantine and isolation, where we're reacting to times when people get sick with viral illnesses. So we're reactive instead of proactive. So can we find ways to be more proactive in the way that we respond to these different challenges that are out there? Okay, so there was a lot of great work done during the pandemic trying to do just that. We sampled in a lot of different places, hospital rooms, gyms, fitness centers, restaurants, you name the type of environment. Somebody was out there doing work and trying to collect airborne viruses from the air and show that, oh yeah, they can exist as aerosols. They can stay suspended for a long time. And so when we did our work, again, these condensation samplers down here at the bottom, you can see have a fairly low flow rate. So if we have something very dilute in the air, if we have our low flow rate samplers, they're pretty good at keeping virus viable, but now we're not able to pull in as many particles, right? And you end up having to run samplers for a really, really long time. It may end up not being feasible to do so. So can we find a way to employ these samplers that help us get, I don't know, a better snapshot of what's actually, or what people are actually exposed to in the environment? So in the work I'm going to talk about for the next couple slides, we used this guy, BioSpot-VIVAS, and it pulls in at air at about 8 liters per minute. And then this guy here, this NIOSH BC251, very common sampler that a lot of folks use for occupational surveillance. The flow rate can be, depending on what you're trying to do with it, 3 to 3.5 liters per minute. And then we've got some swabs that we used for testing surfaces. All right, so we did some work where we went into residences with somebody who was sick and was like isolated to a particular part of the residence, and we found some interesting stuff there. One, we were able to collect with an air sampler virus that could then be cultured. We were able to collect viable virus with an air sampler. Pretty cool. We also were able to, we detected virus, couldn't culture, but we detected it via PCR in this place that was not where the person ever was. And so that raised some questions about how it got there. So we thought, well, maybe we can go into more residences and we can go sample more places, increase our data set. And so we did that, and we figured out from our study that this airborne SARS-CoV-2 similarly present in these rooms where people said they were going to be, in rooms where they said they weren't going to be very often. And so then, if you've got sick people in your home, then you're probably at a risk of exposure to this pathogen. And then our sampler types, right? So if we had our condensation sampler versus our non-condensation, that BC-251 that uses cyclones and filters, there were the sampler type significantly affected that virus collection, conservation of viable virus, right? So in two different instances, we saw these condensation-based samplers performing really well. And so we said, okay, let's go to a medical setting. So we went into this outpatient clinic, and we did some sampling in there, and we were able to detect virus, but we couldn't get our best sampler to where we wanted it to go because it's pretty big and it's noisy, and that tends to disrupt patient care or just staff ability to work in those environments. So we started looking at how do we find smaller samplers that can go into these places that might be better capable of collecting viable virus. So yeah, it's just some pictures from the study. We had staff wear these things, so you could see they wore the BC-251s around, and that didn't bother them too much because we encased it in all this soundproofing foam. But the condensation sampler was relegated to the lobby where the sick patients were not kept too long. So some access issues here. All right, so what can we do with smaller samplers? Well, if you got a smaller sampler, you're probably going to end up with a lower flow rate, presents a problem for using these small samplers in anywhere. So this little guy here, same technology. Here we have our loud BioSpot-VIVAS using 8 liters or pulling 8 liters per minute, and here we have this BioSpot-GEM, same technology. The cooling, the condensing, then the collecting in the particular medium, 1.2 liters per minute. But if we put this thing called a virtual impactor on top of it, then we can bump all that up to effectively 12 liters per minute, which is great because now you're outpacing this guy at the top. So what is a virtual impactor? Well, it's pretty much the same as a cascade impactor. You're effectively pulling your fluid in with your particulates, and you're turning that fluid, and most of your airflow is going away from what is your sampler, where you have a small amount going forward, and you're kind of relying on the inertia, these particles, and this quick turn to keep these particulates moving into the minor flow here. So if you, instead of a collection plate, stick a sampler in this minor flow, now you can pull more particulates into your 1.2 liter per minute sampler, right? And so effectively then you get a 10x concentration factor in your sampler, or the samples that you collect. All right, so that's a virtual impactor. Now, we did not design the virtual impactor, so kudos to the University of Minnesota for their great work putting this together. But here's a breakout of all the different component parts of this device. And so this guy here is an impactor plate, and so this is at the top of this virtual impactor, and it removes big particles. So that's why you see here we have the penetration into the minor flow with this virtual impactor. So you have different aerodynamic diameters on the x-axis here. And you notice that once you get up to about 5 or 6 microns, you're not getting any penetration then through the device and into the minor flow. And in fact, your best penetration into the minor flow is up here, really around 1 micron. And as you get down toward 0.2 microns, the cut size for this VI is about 0.2 microns, right? So you're collecting in this 0.2 to 1, maybe all the way up to about 2.5 microns into the minor flow. All right, so that's a virtual impactor that we used, and we put it on top of the BioSpot-GEM, and this is really a diagram of the setup that we used for our work with what was human coronavirus OC43. So we were running our BioSpot-GEM and our with the virtual impactor and our BioSpot-GEM without it at the same time, so we could kind of compare those two. And you'll notice to run the GEM with the VI, you need a pump that's able to pull those extra 10.8 liters per minute. So we had this guy set up, and so what would happen is we'd supply clean air, dilution air, and then one of these paths would go to our what would be our nebulizer, and that was this Bang nebulizer. So that would send our aerosolized virus, our aerosolized OC43, into this distribution manifold, and then this manifold would send it to the different samplers, as well as our APS, which we were using to measure physical particle size distributions. Okay, and that's not where we started with this. We actually started tinkering around with it using E. coli. It wasn't the purpose of this work to look at E. coli. We wanted to work our way up to OC43, but working with E. coli allows us to work in a less strict biosafety level lab, and so we were able to do our our E. coli work in a setup that looked like this, where we're distributing, we're using a syringe pump, we're aerosolizing the E. coli, and it's passing from right to left through this chamber, and then ultimately being collected by our BioSpot-GEM. We got our VI, our APS, and over here is our BioSpot-VIVAS that we compared things through. Now, it didn't do so great with live E. coli. It did not concentrate how it was designed to concentrate. It's not overly surprising, given some of the unique characteristics of aerosolized E. coli. But if you look at the devices themselves, actually, so this left plot is total CFU. So in our samples, what could we culture? What kind of E. coli could we or how many E. coli could we culture? And this first graph is the GEM without the VI, and the second one is the GEM with the VI. And so you would expect to collect more. The fact that it's pretty much the same indicates that it's probably not working the way that it should work. In fact, this one should really be up above where VIVAS is, because it's pulling more air. You can kind of see that over here when you divide by the total airflow. This GEM VI is much lower, so it's not doing great. But encouraging from this work is that the GEM without the VI and the VIVAS, right, same technology, sampling the same air, they performed comparably. So that's pretty cool. This little sampler performs comparably with the bigger sampler. So that's a good takeaway from this E. coli work. And it was good that we did that, because we couldn't fit the BioSpot-VIVAS on this distribution manifold. It just couldn't support the flow rate. And so we were running the side-by-side with the GEMs once we got to OC43. But on this manifold, we saw good results in that our total OC43, so that is what was PCR detectable. So PCR detectable in the middle there. And then on the right, what we were able to then grow in cell cultures, viable OC43, was at or above the expected concentration factor for the VI, right? So that's good news. The VI was able to concentrate virus. That's a great takeaway from this phase of our work. And then just showed a couple different ways here. This left plot is PCR detectable virus, and this right plot is viable virus. And you see, when you look at the GEM without the VI and the GEM with the VI, you have the GEM with the virtual impactor collecting more virus, right? That's good. It has a higher flow rate. It should collect more virus. But you also see them on par with each other when you normalize for airflow, which means not only are they collecting virus and as you would expect, but they're doing it with a similar efficiency. And you see the same trend over here with viable virus. Total collected and plaque forming units is PFU, and then efficiency here. All right. So we did this work with the BioSpot-GEM and the VI, and this has some pretty interesting takeaways. So remember, I talked about this health clinic study that we did where we couldn't get this sampler in somewhere. Well, there are also other instances, like in schools, where we were limited in the work that we could do because we would be disruptive in these settings, right? So imagine this noisy sampler running in the back of your classroom, a little distracting. But if you could have this quiet sampler, and this one is relatively quiet compared to the big one, then perhaps now you remove that barrier to access to places where you really want to go and assess exposure risks for particularly high-risk populations, right? You can get into these places because you now have a quieter tool to do it. So the fact that we're able to concentrate virus with this VI opens up the door to say, oh, well, maybe now we have a tool that with some improvements could be useful toward better characterizing this exposure risk. All right, so I didn't list a bunch of there are several limitations in the paper and encourage you to go read about that, but there's always room for improvement on these things. And the one, since I've got this giant shh up here, this does require you to use an external pump or some kind of pump. And if that pump is super noisy, then you might, defeat the whole purpose of having a quiet sampler, right? So there can be some design improvements to help make this a more useful tool in the future. But thinking about the future, and I do, this is another, I'm going to ask for your input again. So go back to those Mentimeter displays if you've got them. And I kind of want you guys to think about for a minute here, having heard all this, where do you think the field should go from here? So there, now we got a minute. Everybody's got a minute. So everybody take a break, take a drink of coffee. I'm going to take a little drink of coffee. And this should, as you guys think, from the experiences on this audience, I think we'll have some good feedback here, but it should make a word cloud. I don't know. I guess this just goes to the risk of using a Mentimeter on on this live presentation platform, and I will say I'm also interested to see how these pair up with what we've got queued up after this. All right, automated. All right, automated showing up. Yep, faster. So how can we get them smaller? Okay, 2, 1. All right. Okay, so we've got a lot of good stuff here, and I like the automated bit. So how can we put this thing? How can we put this sampler somewhere that gives us, a buzzer when it detects something? And that's like, when we think about, the chemical side, how can we get some kind of early warning system? I think that's a great thought. And metagenomics. Oh, okay, yeah. So how can we leverage all this new sequencing information? How can we look at, the interesting one is like with bacteria, you can go and you can get some specific markers for bacteria, but is there a way that we can do this for viruses where we can, right now, no is the answer, but maybe in the future, there's a way that we can get to that point where we can take a sample and we can run it through a machine that is looking at some genetic information, and we can figure out if something is a risk without having to go and spend all the extra time in the lab and going through the costly processes that exist now. Yeah. And cheaper is always a good one, right? So and maybe some of these could go together, right? So there might be some upfront costs that we have to work our way through. Think about how expensive it would have been for Pasteur and for Coke to go through with their specialized lab equipment, right? Well, a lot of that stuff is you can order these pre-made kits, right? And there's a lot more accessibility, but to get to that point today, it required a lot of work over time. Can we get to this cheaper point? Yeah. Oh, I love the thoughts. Thank you all for your participation. So what I'm going to do right now is turn it over to Dr. Wu, who's going to go over our future focus area thoughts.
Well, I'd like to thank everyone for your participation. A lot of great ideas, and I know we have some people from the aerosol instrumentation and the detection area, and I think this provides a great food for thoughts of what to do next. And this slide over here is something that I have from my own before we got this input from everyone over here. Size fractionation, which is mentioned earlier, is important. As aerosol people, we often think about, hey, what is a function of a particle size, right? So this will be important. Okay. Can you hear me or no?
I can hear you, Dr. Wu.
Okay, I got a message to say no. Okay, good. Then another one is point of care detection. If we right now, a lot of these samples are brought back to the laboratory for analysis. So if you can do the analysis right on the spot, that will help you to make a decision of the action. That will be wonderful. And our group has started working in these two directions, but of course, there will be a lot more work on this. And then I also like to point out some of these user-inspired development that will tell us what additional functions or criteria requirement will be needed. For example, if we wanted, the examples we have mentioned earlier are more or less for the indoor environment. What if we needed to do it in the outdoor, very open environment? Let's say we want to collect viruses from the crop field or from the ocean. Then this simple device that we have come up with probably will need to have a scale up. And the next one is a lot of people are working on collecting bioaerosols from biomass burning and wildlife. Yes, even with a high temperature, they are still there. And at the same time, what will be the requirement for a sampler that is able to do that? In another direction, a lot of people are working using on drones to collect samples, right, at different altitude, different height, and gives you a better spatial distribution. But we don't have such a small sampler that can be used for drones. For flights, yes, but for drones, not yet. So that's some area that requires improvement. Okay. So can you move on to the next slide? So this is what we have for today. And of course, we like to acknowledge the financial support from agencies, NIH, NIOSH, SBIR, and U.S. Army. And also wanted to also appreciate all the people who have participated in this research. It's not possible for one person or two people to get all these done. This is really a great teamwork from all these people together. And so with that, we'd like to conclude today's lecture. And thank you for participation. We'll be more than happy to answer any questions you may have. Thank you.
Thank you so much, Dr. Chan Yu and Dr. William, for that excellent and insightful lecture. So now we'll open the floor for questions and answers. You're welcome to drop your questions in the Zoom chat, which I'll read it out. So feel free to drop your questions. So I think I have a first question which says, since VIs have a cut point for which to work, what cut point would be reasonable given that viruses are always attached to particles? Is 1 micrometer sufficient for effective sampling? This would greatly simplify the design and operation of the VI.
Yeah, this is a great question. So I think for this work, the cut point was designed to be 0.2 micron because there is not much below that. But according to past studies, the distribution of viruses is quite widespread, depending on the type of viruses, and they still exist in the sub-micron range, and therefore 1 micron you may lose some, and that's why for this work it was designed to be around the 0.2 micrometer.
All right, thank you. Then the second question says, have you made acoustic measurements with a VI on the sampler?
I will let Brett answer this question because he was the one who had experienced the noise.
Yeah, no, we did not actually measure decibel levels. It would be a good thing to measure, though. Could be done very, very quickly. We just didn't have the meter available. But that's a good question. I'll tell you, it's definitely louder with the pump on, the 10.8 liter per minute pump on than without, and that the VIVAS is noticeably louder than the BioSpot-GEM, that smaller sampler.
All right, thank you, Brad. The next question is saying you have a huge flow rate from the drone's propellers. While the cut point will vary and you might have trouble with quantifying sampling volume, can you put an impaction surface right on the props?
So to put an impaction surface, I would say that may be an interesting idea. I actually like to see if there are inputs from the audience to help out with that, who have more experience with the flight or drones environment.
All right, thank you. Then another question is why did you choose an APS instead of an SMPS?
Yeah, no, that is a great question, and it would have been a good idea to have an SMPS attached also. We did do a small number of samplings with a SMPS, I don't know, equivalent in terms of particle size range measured, but we weren't able to use that given the flow rate on the distribution manifold and how many different things we needed to sample. And we also just had a equipment limitation with multiple ongoing different projects. And the SMPS we did have was elsewhere, and we didn't want to be trying to move it into the BSL-2 environment and out of the BSL-2 environment to support both of those projects while also being concerned about the disinfection of the device itself. But you're right. I mean, so in one of the limitations that we do have in this setup is that we know because of that sampling that there were a lot of submicron particles that were produced in the manifold itself. And so even though the APS can't pick those up, we know that they were present, and so there's some impact on the sampling. We don't know exactly what it is, but certainly it would require further investigation.
All right, thank you.
And I did, if I may, I mean, I did have a thought, and as Dr. Wu said, I think this would be a great one for folks as a community to think about. But how do we overcome the downdraft from those drone propellers? I would imagine some kind of device that extends up above where that downdraft is coming. But having no experience in doing that, I do find it a very interesting topic that it would be good for. I'd enjoy hearing folks from the community talk about, if not here, other places.
All right, thank you. Then another question is, have you tried to design a VI for the VIDS to enable higher flow rates? And were there any obstacles doing that?
That's a good question. I believe University of Minnesota has been working on that. I don't know if anyone from Minnesota is in the audience that may want to share. we haven't done that ourselves.
Yeah, and to answer Ari's question, that is where the VI came from the University of Minnesota.
All right, thank you. The next question says, where did he get the VI from?
So it's part of a project with the Hendix, previous aerosol devices, and with a collaboration with the University of Minnesota. So if you are interested in getting one similar one, I would suggest you to contact aerosol device, Hendix nowadays.
All right, thank you. Then another question says, what was the size distribution of the OC43 virus?
Yeah, that's a great question. So that is actually, it is in the paper, but let me try to share a screen here. This one wasn't in the slide deck, but do you guys see my screen? There it goes.
Yes.
It's back to that lag again. So when you ask what the size distribution is, that can be a tricky question because there are multiple size distributions. So you've got a number distribution, which is this, and I'm colorblind. I should have picked better colors. But this line here is your number distribution. Then you've got a volumetric distribution, and then you've got a surface area distribution. And so from other work that was done with this setup, we determined that the surface area distribution was the one that best correlated with the collection of OC43. And so the surface area distribution you can see here in what I'll call blue, but who knows, it could be purple. And I'm not sure if that answers your question, but you can see you've got a peak here, a little less than 1 micron. Does that answer your question?
I wanted to clarify, this is the physical site distribution measured
yes.
by APS. Yes. Yes.
That's a great point.
Now, if you are asking us the distribution of OC43 in the particle, then that's another topic to work on because you do need to have the right tool to get this distribution of the viruses as a function of particle size, and that's not an easy task.
All right,
Thank you for that clarification, Dr. Wu.
All right, thank you. I guess one of the last few questions, do you have a guess of what aerosol size range was carried by the bulk of the virus?
I would say for this study, then that would be probably from low microns down to some micron, but that depends on the nebulizer you use, right? So for this, we use the band nebulizer. Now, laboratory generated virus aerosol is not going to be the same as you have in the field when you go to sampling in the student healthcare center, in the hospitals, in the restaurants. And these are produced by human beings. They have a different distribution. Oh, so that will be a different story. So my point is, don't directly translate what you learn from the laboratory nebulizer generated bioaerosol for the actual field distribution.
All right, thank you. If there are no other questions, I think I have a last question. So talking about field sampling, has the effect of humidity been considered on the instrument performance, when sampling the field?
Sorry, say it again.
like the effect of humidity, on the instrument.
Oh, effect of humidity?
Yeah.
Yes. Brett, do you want to take on that first?
Sure, I'll take a stab at it. I mean, the humidity, the relative humidity is going to impact particles in the air, right? It's gonna drive how quickly the liquid, the moisture from your respiratory system evaporates. It's going to affect how long particles stay suspended. And so there's a little bit of a give and take when it comes to relative humidity. The quicker that the water evaporates, a dry environment, the smaller the particles get, the longer they can stay suspended in the air. But also, the quicker that moisture evaporates, the less protected viruses may be from, particularly like pH changes in the droplet itself as it's leaving your respiratory system. So, I think that relative humidity should be something that is measured when you're doing sampling like this, so that you can correlate. But in general, you see like this. It's kind of a U-shaped curve, right? Because in terms of viability, whereas the viruses are more protected, then they are able to stay viable longer. But you might be able to collect more viruses when those particles can stay suspended in the fluid in the air for longer as well. Just how is that, Dr. Wu?
Yeah, wonderful. Yes. And at the same time, I also have one supplemental point. That is, most of the time we evaluate the effects of a humidity on viruses in the laboratory environment, right? So again, these virus aerosols generated in the laboratory may not be the same as you would encounter in the real field. So the rate humidity effects will also be different. Now, if you are going to do sampling in say inside buildings, then most of the time I think you have the real humidity and the temperature control because of the air conditioning, right? Now, if you go to do field sampling outside, in crop field, in ocean, in the forest, that will be very different. So that's a important point that you needed to monitor and study their effects, but I don't know how much we know about the effects of real humidity on the open environment yet.
All right, thank you. So I'll just take this last two questions. The first question says, what sampling times do you think was the sweet spot for the virus detection with this flow rate of 12 liters per minute via the system?
Yeah, that is a great question. And unfortunately, when you're doing these samplings and whatever environment, pick your environment, the environment is often going to dictate how long your sampling times are. So like for an occupational study, maybe somebody can wear a sampler for 8 hours, and you let that sampler run because you want to characterize their exposure over the entire workday. But maybe in a classroom environment, classes change every, I don't know, hour. And so you're really going to run, you might want to run every hour and change your samplers out. So I would say the sweet spot for virus detection is pulling in the most air you can while not stressing out's the wrong word, but not inactivating what you're collecting, right? So it's finding the right type of sampler, maximizing its potential in terms of its flow rate, and then employing that as effectively as you can in whatever complicated environment you find yourself in, and trying to find the most rational reason possible for why you chose X time in a certain environment. So I don't know that there's a sweet spot for, in an ideal world, you might be able to like turn up the sampler flow rate so that you, no matter the time, are collecting the same volume of air. But it's very situation dependent. What would you add, Dr. Wu?
Yeah, I agree. At the same time, I wanted to share a little bit more information about the function of time. So for this specific sampler, the BioSpot-VIVAS, we have done in the laboratory the viability of the collected viruses, and 10 minutes, 20 minutes, 30 minutes, there was no change. So that's a proof that the BioSpot works very well for sampling the viruses and conserving their viability over the sampling time we have tested. And that Now, if you are using other samplers, then I would recommend lower duration, because some of these collected viruses, let's say on the solid surface of say filters or cyclone, then you may not want to do the sampling for a long period of time. For the short period of time, the viable viruses may still be live over there, but long duration, that a lot of them just get inactivated.
All right, thank you. Another question I have here is, I think it's like 3 questions in one. Is there any data? Is there any data on the size distribution of the viral particles in realistic environments? Then also, how well does this compare to like the lab measurements? Then also, to what degree are there viral clusters rather than individual viral particles? And also like viral particles mixed with other materials. Then if the particles are individual viruses, how infectious are they? And is there an infectious threshold level, of the SARS-CoV virus?
Oh, there are a lot of questions.
Those are good questions. Those are good questions. You want to go first, or you want me to?
Well, in terms of the size distribution, now if you just talk about the RNA, and there are a lot of studies, especially during the COVID time, right? For SARS-CoV-2, people collected them as a function of size and use the PCR to determine the RNA. And you can see the distribution almost everywhere, depending on the studies. They are in the submicron, but they are also in the supermicron. These are the real samples from the field. Now, in terms of the viable one, then that's a little bit more trickier, because you do need to be able to conserve. And I think there are John Lenicky, they did some sampling using a cascade impactor for passengers in a vehicle, and you can see I think the majority of them are in the say 1 to 2 and then the submicron, and not really a lot big in the higher size, because maybe because the water would evaporate, so the size after the water evaporation is getting smaller. So that's what I remember. But there are a lot of studies on the distribution of the viruses in aerosol particles in the real field. But most of them are based on the RNA, limited on the viability one.
Yeah, and I will add that you run into a similar problem that was discussed earlier when Dr. Wu corrected me on physical particle size distribution versus OC43, right? You may have to do these measurements of physical particle size distribution and your sampling with whatever sampler you have at the same time, and it's often hard to do that in a outside of the lab situation. So there are a lot of studies that talk about the collection of virus from the air, but you end up with the same, the information is not correlated with physical size distributions. And I will, let's see, what was the second? Are the viruses together as clusters rather than individual particles? Are they mixed with other? Yeah, they're always mixed with other materials. And in fact, that mixing is what often makes the lab versus real world difficult to layer over each other, because you make your medium in the lab and you try to make it look like something in the real world, but at the end of the day, what somebody's coughing out of their respiratory system is very complex, and so there are other things in there that are often protecting that virus from inactivation as it goes out into a harsh environment. So those viruses, they can be individual things, but they're most often part of another particle, which is why they can show up in those different size ranges. And how infectious are they? I'm going to vary, and of course it varies by pathogen also. But for SARS-CoV-2 in particular, you only need a handful of virions, infectious viruses, to transmit disease to a susceptible host. So you don't need very many of these things to get people sick. So if you need to collect a bunch of them from the air in order to detect them, you might be missing a lot of the potential exposure risk, which is why we need to get these samplers able to detect lower and lower concentrations in the air.
I also wanted to point out that yes, so in the field, say in hospital, these respiratory viruses are produced by human, coughing, breathing, all these things. And saliva is an important component in some of these. And in the laboratory, we can also use saliva as the suspending media and to aerosolize, these saliva-containing viruses. But how close are they to the actual field? I think that's another story.
All right, thank you. I think we have time for one last question. So the last question is, in your experience, what is the maximum practical duration of sampling for the VIVAS?
I'll go. So for VIVAS, actually this answer, we'll see what Dr. Wu says, because this answer might have changed since I left the program a year ago. That's how quickly it develops. So your limitation is really how long can you keep liquid in this petri dish that the virus are being collected into. And so we run those samplers for like 8 hours or so, but we would run them most often we'd run them for about 4 hours, and we do 2 a day. And thinking about humidity and challenges to overcome, with these types of samplers, you're fighting whether or not you're going to evaporate all the liquid out of the sampling well or overflow it. And so you've got to dial that in and adjust temperatures on the device itself. And what was being developed when I left the program was a way to with imaging detect what the level of the device is in this collection well is, and then adjust different temperatures in the device to keep that level at the same rate, where theoretically you could run it for as long as you wanted. But yeah, we were about usually 4 hours, and then we changed the sample. Dr. Wu, did you want to add anything to that? Or
yes, so typically we run for 3 to 4 hours, and you can run it for 8 hours depending on the liquid level, as long as you can monitoring. So we're still working on that, and I would suggest that for people who are interested in this, actually contact Hendrix and Aerosol Dynamics, because they are the ones that we work actively in getting this design and development done.
All right, thank you, Dr. Wu and Dr. William, for creating time, to present such an awesome lecture, and also thank you, Amy, as well, for organizing this lecture. So we've come to the end of the July lecture series, and looking forward to seeing everyone at the August lecture series. I think is there any? Okay, all right. See you, everyone. Thank you very much for joining, and have a great week ahead.
Thank you very much.
thank you all.